was asked to introduce Jared Westbrook. He's the quantitative geneticist for the American Chestnut Foundation. He has his master's in botany and his PhD in plant, molecular, and cellular biology from the University of Florida. And I hope you're all ready to be blown away by science.
So what motivates me to work on chestnut is that it's an interesting example of a tree species that's been wiped out by this exotic pathogen. And we, as the American Chestnut Foundation, has progressed in the last 30 years. We're now on the cusp of actually bringing this tree back, and it would be an unprecedented thing to basically take a tree that's extinct and bring it back to life. And we're using a lot of different technologies in parallel to do this. One of the first technologies where I won't talk much about this, but there's hypovirulence, where a virus infects the chestnut blight fungus. And you can see, these are the plates of the chestnut blight fungus. And you can see the plate where the hypovirus has infected the fungus, the growth is less. So the fungus has reduced its pathogenicity on the tree. And right now at the University of Maryland, the reason this is not used widely, why it's not spread out among the range of chestnut, is that in order for this virus to be transmitted between the different strains of the fungus, the fungus need to be able to merge their hyphae together and to transmit this virus. And so there are certain strains or genetic strains of the fungus that they are not compatible to merge together like this. So at the University of Maryland, they figured out which genes in the genome of this fungus are responsible for making these different strains of the blight fungus incompatible for mating. And they've knocked those genes out, they've mutated them out of the genome, so they're no longer functional. And so they've created a super donor strain of this fungus, which can mate with every other kind of, or merge, I guess, with every other type of fungus out in the forest and transmit the virus to it. And so this would be one, they're still going through this process of seeing if this is a safe technology. They have to go through the regulatory process with the government. But we could potentially apply this super donor strains of the fungus to different cankers of the blight, and that would transmit the virus and make the blight less pathogenic. The second thing that's been going on for the last 25 years is transgenics, and I'll talk more about this later, but basically at the State University of New York, they found a gene from wheat. They inserted it into American chestnut, and when the American chestnut expresses this gene, it becomes much more blight resistant. You can see they've taken the original tree where the gene inserted, and they've crossed that tree with other Native American chestnuts, and the progeny of those crosses, half of them inherit the gene, half of them don't inherit the gene. And you can see the difference in resistance very clearly. This tree inherited the gene, this tree did not inherit the gene. And then the third I'll talk about more is backcross breeding, which is what we're doing, what the Massachusetts chapter is doing. That's basically to recover to hybridize the Chinese chestnut tree with an American chestnut tree and backcross it repeatedly to American chestnut to recover the American chestnut type. So with the backcross breeding, you're starting with this Chinese tree that's resistant and American chestnut that's not resistant. You create this, what's called the F1, so half American chestnut, half Chinese chestnut. These early breeding programs started in the 1920s, created these hybrids. And they found that the hybrids had intermediate blight resistance and they didn't grow like American chestnut. And so this guy in the 1980s was doing breeding with corn, and he knew about this old style of breeding, backcross breeding, where basically they take these hybrids and they cross them repeatedly to American chestnut to recover basically a tree that's essentially 94% American chestnut. So you get to this third backcross stage. These trees are going to be intermediate in resistance because every time you cross an American chestnut tree with these hybrids, you always are getting that susceptibility from American chestnut in combination with the resistance from Chinese chestnut. So in order to combine create trees that have resistance from both parents, you have to do this crossing between these third backcross trees that are the most resistant in this population. You get these B3F2 trees. And then every time you're doing this crossing, you're rolling the dice basically. So if there's 3 genes that are involved in resistance, 1 in 64 of these trees are going to have inherited all of the genes for resistance. There's a whole spectrum of resistance that's in that population, from 0 resistance to almost full resistance. And so the breeding program was started by basically these 2 trees. There are 2 founder trees, the Graves tree and the Clapper tree. And this guy, Fred Hebbard, took American chestnuts from Virginia, and he crossed them with these Graves trees and the Clapper tree, so 20 different Americans on these single trees. And created these backcross lines, and that's what this is showing. So this is like the original Graves tree, that's the original Clapper tree. And you create these families. There's 20 different families, and each family, they had like 100 trees in the family that they selected from. And they selected about 2 to 3 individuals from each of those families that are resistant. And then they took another set of 20 Americans, crossed them with the best trees from these families and the best trees from those families. And so you end up with this third backcross trees. And that's basically to recover some of the diversity from American chestnut. And that's now been expanded out to the chapter programs. So pollen from the best backcross trees. We've sent you pollen from these trees. We have given it to all the chapters, and they've crossed that pollen with American chestnuts that they found flowering in the forest to basically capture some of the diversity that's remaining in the forest. You can see that for these major sources of resistance, we've created about 260 different backcross lines, or 360 different backcross lines. And so all the way throughout the range, this has been a citizen science effort to basically recover some of this locally adapted material. And you can see in Massachusetts, Rhode Island, there's 40 different Clapper and Graves lines. So you might have seen these kind of charts in introductory genetics, where you take these backcross trees, which these letters indicate, the big letters indicate whether the tree has the resistance gene, and the little letters show that the tree is also susceptible to, has a gene for susceptibility from American chestnut. So if you assume there's 3 genes for resistance, and you cross a tree that has, there's intermediate resistance at all of the 3 loci, you're going to have to screen through what this graph is showing basically, the main point I want to make is that 1 out of 64 trees approximately will be fully resistant. We'll have inherited 3 copies, or 2 copies of each resistance gene from both parents. So that's why you're seeing in Brian's slides, all those chestnut trees planted all very close spacing. You have to screen through many, many individuals in order to get the resistant individuals. And so we've been planting these seed orchards. You plant each backcross line in 9 blocks. So every one of these lines, you plant out 150 trees 9 times. So we end up planting 30,000 trees in these orchards, and we select 1 out of 150. So we end up going from 30,000 down to 250. It's an incredible, massive effort that's been going on, and we're further along in our breeding program in Meadowview and calling these populations down, but we still have a lot of work to do to get to the point where we're down to that 250 trees. So what we've been doing is artificially inoculating these trees with a chestnut blight fungus. And you can see that some individuals demonstrate like really big sunken cankers. And those trees are highly susceptible to the blight. We get rid of those trees. We see some trees with intermediate levels of resistance, swollen cankers. We also get rid of those trees, and we keep the trees that have these smallest cankers. So we end up killing about 80 to 90% of the trees just based on the growth of the cankers. That doesn't really get us to the 250 trees. So if you think about the math, we're talking about 60,000 trees we planted in both the Clapper and Graves orchards. We're down to 10,000 now, and we want to get down to 250 or 600 total about for both sources of resistance. So we still have a long ways to go. There's still plenty of susceptible trees in those orchards. So so how do you get, so now we can't really tell. We got all trees that look like this. We have small cankers initially. Some of them look pretty bad and we can get rid of them further based on how they grow and get to blight later. But we have to go through and screen these trees that remain. So we have this unselected seed orchard with 150 trees in our block. We get rid of the worst of the worst. So now we're left with this smaller population. And we want to get down to like the best tree. And so we have these partially selected orchards. We allow these trees to intercross with each other, and we generate progeny from these trees. And we inoculate the progeny with the blight, and we infer the resistance of the parents based on the average canker sizes of the progeny. So here you can see in this graph I'm showing different families of trees that originate from these different mother trees. And we're trying to find the families with the smallest cankers. And we're going to select that one individual. So you could imagine we're going through this these seed orchards, and now we have, like, let's say 10 trees in a plot. For every one of these 10 trees, we have to plant out like 30 to 50 of their progeny. So now we're blowing up the scale of the problem to make it so we're planting thousands and thousands of trees to be able to screen through and find the best one. So it's a pretty slow process, and I'm not very patient, so I want to learn ways to make this process go faster. But what we're seeing in these progeny tests is that if you look at American chestnut canker sizes, this is the American chestnut canker sizes, these are the Chinese chestnut canker sizes. These are the canker size variation of the progeny of the trees that are remaining in seed orchards after we've done our initial culling. We find that the trees, the best of the best, are about intermediate in resistance. They have the average canker sizes in these families. So there's probably going to be individuals that have resistance that approaches Chinese chestnut, but the average of the population is not going to probably reach that of Chinese chestnut. The idea was to get to Chinese chestnut levels of resistance. So why is the breeding program falling a little bit short? Even though we do have resistance, why is it not to the level of Chinese chestnut? One hypothesis is that we had originally thought that we were always selecting for these same 3 genes in every backcross line. But there could be more genes involved than 3. And the American chestnut backcross lines, when you go through and inoculate in the breeding orchards, you're selecting based on the site of the cankers. So some of the trees, it's not the perfect resistance assay. It doesn't really tell you, just looking at the cankers doesn't tell you actually how many genes for resistance a tree inherited. So these trees there's a strong environmental effect on the growth of the cankers as well. And so that environmental effect might distort our selection process. And there might be more genes involved in resistance. We have this variable inheritance of resistance genes in each of these backcross lines. And if you look at this graph is kind of complicated, but these are the different backcross lines from our breeding program in Meadowview, Virginia. And these are the average canker sizes with a variation. And so we see in the backcross lines, if we expected like perfect selection, every one of these backcross lines should have equal average canker sizes. But in fact, we see some lines that have bigger than average cankers, and some lines that have less than average, smaller than average cankers. And so what I'm suggesting is for your seed orchards, rather than planting, going full out and planting 9 blocks right away without knowing the results and spending a lot of time on these trees, I would suggest planting fewer blocks, figuring out these guys, figuring out the best lines to advance, and then spending your time on that material. Plant more of that, plant less of this, and get rid of this material so that they're not contributing to the susceptibility in the population. We also have a second pathogen, Phytophthora cinnamomi. This infects the roots of chestnut trees, and rather than, the trees just die from this disease. They don't just even resprout. So in the areas of the south, in particular, the coastal plain of the south, it's thought that this disease wiped out American chestnut prior to the introduction of chestnut blight. So there's been some screening for Phytophthora resistance in our breeding program. And so we've been infecting these trees with rice grains with this root pathogen. And we find that some of these trees die and some of them have resistance. And we're able to tell which mother tree has the most resistance based on the mortality, the average mortality of the progeny of the mother tree in the seed orchard. And so what I'm showing is there's a strong evidence that this Phytophthora cinnamomea resistance is controlled by genes, by a few genes with very large effect. So we've done no selection previously for Phytophthora resistance. We've been screening for blight resistance for the past 30 years, but these advanced generation hybrids, about 80 to 90% of the variation among these families is due to genetic variations in the mother trees. So it's even though we've done no selection, we have a very strong signal, genetic signal coming out, and there are some highly resistant families in this, particularly this Graves source of resistance. And I'm showing you here in this slide relative resistance of American chestnut, Chinese chestnut, and Graves. And you can see that the Graves trees, even though we've done no selection for resistance, these trees have resistance that approaches Chinese chestnut. So that's very hopeful. But still, we don't really know what's going on at the level of the genes. We've just been breeding trees and selecting for resistance based on how they look. So it's time to figure out what's going on in the level of the genome. What is the genetic basis for resistance in TACF's breeding program? So we have this 3-step process where we're going to do some genomic sequencing to understand the basis for resistance. And the first step is to sequence a reference genome for a Chinese chestnut tree, an American chestnut tree. And basically what it means to do a reference genome is to take an American chestnut tree, extract the DNA, do some sequencing where you create you create these pieces of DNA, and then you assemble them all together like a puzzle. You're trying to put them all together back into their chromosomes. So it's like a parts list. So you don't really know what the genes that are causing resistance or susceptibility, but you know the parts list, so you can actually figure out which genes are controlling resistance. The second step is to go to the BC3 individuals. Those trees are 6% Chinese chestnut, 94% American chestnut on average. And so you figure that the reason those trees are resistant is because they have some genes from Chinese chestnut. So we can figure out which parts of the genome in these BC3 trees, this red is indicating the Chinese tree and this blue is representing the American tree. We can figure out by sequencing which parts of the genome came from Chinese chestnut. Those islands of the genome should harbor some resistance genes. And it's possible that the different backcross lines inherited different segments of the Chinese chestnut genome. So we figure out where the Chinese genome is inherited. And the third step is to basically do a scan of the genome from these individual BC3 F2 trees that basically go from 0 resistance to almost 100% resistance. You scan for correlation between canker size variation in this population and DNA variation in the population. And hopefully what we're going to find is that there's a strong correlation between resistance and the Chinese chestnut segments of the genome. And then we can figure out a genetic test and basically just sequence the DNA in those regions that are controlling resistance and be able to tell if the tree inherited the gene or not. So recently we've gotten money to do reference genome assembly for American chestnut. So we got $100,000. We're partnering with this Hudson Alpha Institute for Biotechnology. We're funded by the Colcom Foundation in Pittsburgh. And so we're going to sequence it's called the LS1 tree. It's one of the American chestnuts that's used that has the gene from wheat inserted into it that enhances its resistance. And so it's cloned, it's resistant, it's not going to die. We can clone it out, and so this tree is a useful reference that will be bred with American chestnuts from throughout the range. So I want to start leveraging that reference genome to do some other interesting studies. And one of the things I was going to suggest to you guys today is within your chapters, you spend all this time creating these backcross lines. You spend a lot of time doing selection within them. So 20 graves, 20 clapper lines. We're talking maybe you selected 1 to 3 individuals per family in those lines. For about $100 a sample, we can basically genotype those selected trees and figure out which parts of the Chinese chestnut genome those trees inherited. And so what I'm asking is for you to help with fundraising of about $5,000 to $10,000 and help us collect some tissue from those trees. We will genotype them for you and tell you which portions of the genome came from Chinese chestnut, and you can figure out, are your backcross 3 individuals, did they inherit the same Chinese chestnut genes and same genes for resistance, or different ones? And if they inherited different ones, it should inform your breeding to determine whether you can cross those trees with different resistance genes and enhance the resistance in the next generation. And so the idea is to sequence these trees, and what I'm doing for the MetaView breeding program is basically only sequencing the genes. So we sequenced about 25,000 genes for $100 a tree, and we sequence the genes and we compare it back to the Chinese source of resistance, like the mahogany tree that was the original founder tree for these populations, and we figure out which genes were inherited from Chinese chestnut, which genes were inherited from American chestnut, based on just comparing the sequences of those genes. So that's one thing that I think the chapters can help with in fundraising. And each chapter, the other valuable thing that this will contribute is to understand what is the component of American chestnut diversity in the breeding program, and how much of the diversity of American chestnut that's out in the forest have you captured in the breeding program? Are you representing all of the adaptive genetic diversity that's important for restoring the species? Or is there more diversity out in the forest than there is in the breeding program? And if there is, we should go and get it with some germplasm conservation. It will help us to identify where we should go and collect more seeds so we can do more germplasm conservation for diversity. I'm also working on developing this genomic test to alleviate the need to do this very laborious process of taking nuts from every individual mother tree as a candidate for selection and planting out the progeny and figuring out how resistant the mother tree is. That's a very slow process. What I'd like to do is be able to go to the mother trees that are remaining in the orchards, pull off a leaf, sequence DNA, and say, does this tree have all the genes for resistance, or does this tree have less than all the genes? And if they have less than all the genes, let's get rid of it and get only the best trees for further breeding. So the idea with genomic selection is you basically pepper the genome with markers. So you don't sequence the whole genome of these trees, that'd be too expensive. So you get these markers scattered throughout the genome, and you have a gene that's involved in blight or phytophthora resistance. These markers are in the neighborhood, and the markers sort of capture the effect of those genes. And so you associate the effect of the individual markers in a statistical test with the variation in the resistance in the population. And through that process of doing the association between the DNA sequence variation and the blight resistance, you're able to develop a predictive model that you can just sequence DNA from the trees and be able to determine whether the tree has resistance or not. The other reason for doing DNA sequencing is to understand what's important for adaptation in chestnut. So we can also go to wild American chestnut populations and sample contrasting environments, let's say high elevation and low elevation. And we can see whether there's certain gene variants, these are obviously depicting the gene variant very simply as with a red and a green, whether there's certain gene variants that are associated with, let's say, high elevation habitats. So that might be indicative of some kind of natural selection for high elevation traits for this particular gene in the genome. This one here, I'm showing that there's no association between this high elevation or low elevation and high elevation. So we scan the genome for these adaptive associations, and we look for, I guess, rare populations that harbor adaptive diversity. and we go out and we conserve that, we bring that into an orchard for conservation. So I'm also suggesting in the next season or so to go around Massachusetts and Rhode Island and collect a dispersed sample of American chestnuts. And I need them all to be geo-referenced leaf samples, so I need the coordinates. So find a dispersed sample, like 50 to 100 trees from throughout the range. We're also working on mapping out which trees we use for backcross breeding. So areas maybe that we haven't used for backcross breeding, try to sample those areas. Send the tissue to me in Meadowview, Virginia, and we're going to try to find some money to do some sequencing on these trees to understand what is the important component of adaptive diversity. So we'll try to do this for Massachusetts and Rhode Island, but we'll do it all the way from Alabama to Maine. And we'll associate climate variables like precipitation, average temperature, with this DNA sequence variation and scan the genomes for these important adaptive diversity and find pockets or regions of adaptive diversity that we want to go out and conserve, spend our time on. So those would be put into germplasm conservation orchards. So we would find the trees, we would try to find a mother tree, collect pollen, collect seed, and pollinate try to get as many American chestnuts into these orchards that we can use for future breeding. So the uses would be for long-term conservation, research, educational opportunities, more understanding what's adaptive variation in these populations. So like taking American chestnuts from Maine, from Alabama, planting them in a common garden, and seeing what are the traits, how do they vary? Like, do the flowering time vary? Does the cold tolerance vary within the same environment? And so this will be, this is the first step, is finding the trees and putting them into an orchard, into a common garden. And then to diversify the transgenic tree. So this transgenic is very exciting. So basically they've taken a gene from wheat. This is, it's called oxalate oxidase. And the fungus produces this metabolite called oxalic acid, which is part of the reason that causes the cankers. So this oxalate oxidase gene basically detoxifies this oxalic acid, turns it into this CO2 and hydrogen peroxide. And trees that have the gene, you can see this is a tree that has the gene. They're doing great they've been inoculated with a very pathogenic strain of the blight. This is a wild American chestnut that's wilting, and this is a Chinese chestnut tree that's also wilting. So it even seems to be doing better than Chinese chestnut. This is a gene from wheat, but it's found in banana, rice, barley, sorghum, strawberry, all these things that you eat all the time. So it's not a gluten type of enzyme or anything that's going to be dangerous for you to consume because you consume it all the time. And I guess what I like about this from the point of citizen science is that it's a lot easier to, if you're going to have a single gene that's going to confer resistance, half of the progeny are going to inherit this gene, half are not going to inherit it. So it's a lot easier to screen for than planting out thousands of trees and picking one from those thousands. You pick out half of the trees. It's pretty easy to detect if the trees inherited the gene. And you don't need a PhD, and you don't need to do genomics or anything like that. You just have an enzyme assay and you determine whether the tree inherited the gene or not. So I think it's good. It has a lot of potential for citizen science once it's deregulated. They have to go through this process of seeing whether the insertion of the gene affects relationships with beneficial pollinators, with mycorrhizal fungi, does it affect the growth of the tree? So they're going through this whole process of seeing if it's safe, and then that will be determined whether it will be released by the U.S. government, the EPA, the USDA. So that they're still determining that. It should be about 3 to 5 years. So we could integrate this transgenic blight resistance into the breeding program, and basically what it's going to take is going to find wild-type American mother trees and pollinating with the transgenic pollen once it's available for release. So like I said, one half of the progeny are going to be like this, not inheriting it, and one half will look like this, which is highly blight resistant. There's so much variation in the species that you can see that there's like a definite trend towards certain variants being more abundant in Chinese and certain variants being more abundant in America.
So here in the Mass Rhode Island chapter, we also have the 10 or so lines of Nanking, and the South Kingston orchard, of which there'll be a tour in a little while, includes one of those Nanking orchards. It's had some soil problems, so we're still, and it's autumn anyway, so you wouldn't see the leaves. But the thing that I remember hearing about Nanking is that it may have retained on somewhat more common basis some of the Phytophthora resistance. Do you happen to know, and can we have available the Phytophthora ink disease resistant lines so that we're sure to pollinate any future mother trees with
What I've
such
seen in the
lines?
data is we've progeny tested some of those Nanking selections at Metaview for Phytophthora resistance,
Yeah.
and it looks like every generation, like the average Phytophthora resistance decreases. Like we're diluting out the Phytophthora resistance through back crossing. We got lucky with grapes that we didn't dilute it all out, and there are some lines that appear to have some intermediate level of resistance, but I'm not really optimistic that the Nanking advanced backcross trees have much Phytophthora resistance. Definitely Nanking Chinese does,
Yeah.
and there are earlier generation hybrids that we have that may have that resistance in it. So but I have to go back to the data and tell you which ones
Okay,
are
so-
promising.
So we should maybe find that out then,
Yeah,
because
I can tell you which graves lines.
yeah, okay,
Um,
yeah, and just to be, so everybody who's working in Mass Rhode Island knows, currently our chapter is only working with the conventional back cross breeding, and we have a variety of attitudes on, and opinions on transgenic technologies represented in our chapter, and so but we all do agree that we want chestnut to survive long term, and there are different ways that the programs could be combined. Anyway, it doesn't have to occur in our states. We certainly might want to keep the orchards purely conventional.
You can keep your orchards conventional, and you can
Yeah.
also plant germplasm
Yeah.
conservation orchards
Yeah,
now.
yeah.
I would suggest doing that now.
Yeah,
And for the purpose of crossing with the transgenic for those that want to do that.
yeah.
If you don't want to do that, continue with backcross
Yeah,
breeding.
so there's
But there's room for
People don't
everybody
have
to do
to.
their thing.
yeah.
And yeah,
So, two things. We have all of these backcross BC3F1 orchards that we're now using and planting and creating seed orchards. And we think we've got a rough idea of the level of resistance. And I was listening to what you're saying. Are we at a point where we could test those trees that are selected and pick the ones to be able to do a leaf assay or some other way to say, it tested well, but it really doesn't have the resistance we're looking for, and get rid of them at this point?
the BC3F1 trees.
Yes, for the ones that we've already selected for our backcross orchards, which we're now producing seed. Is there a way to test those trees? Do we assume that they have a moderate amount of resistance?
What I would suggest is plant your seed orchards, inoculate the seed orchards, 3 blocks minimum. Then you know how resistant the mother tree was based on the average canker size of the progeny relative to the other mother trees you have. It's
So-
like every generation is a progeny test, basically.
Right. So, what I'm leading to is that we have this third line of resistance,
Yeah.
that we're not gonna have a full 20,
That's fine.
full 20 lines. But I think of it like as a multiplier, where it can be crossed in various ways with the other 2 lines of resistance. And being able to select those with a leaf assay, instead of having to plant them out as seed orchards to figure out how to do that, is that something we're close to, where we can?
Combined resistance sources, is that what you're asking?
Yeah, use the Nanking line of resistance as to up the level of resistance of the other two without having to take the Nankings and do an entire seed orchard. Are we close to that?
If you have the Nankings planted in another orchard, the breeding orchard, then the Clapper and Graves, then you're only going to get Nanking by Nanking crosses unless you want to do CP or controlled pollination, which is a huge effort to generate the number of seeds required for a seed orchard.
True, but what we want to do is select the most resistant ones among the Nanking and then cross them. So what I'm looking for is ways that we can, select those faster without having to go through the laborious process we're in right now. Our biggest challenges in the chapter is that there is so much work to do and not enough people to do it. And that's our biggest challenge. So ways that we can save labor.
I would say try the small stem assay. Try the seedling inoculations and see if one thing to consider is to make sure that the trees are big enough. So get nice, hardy, robust seedlings. Plant them out early as possible in the greenhouse. If you have a greenhouse, or cooperator with the greenhouse. Inoculate them in May, June with a weekly pathogenic strain. Find the worst ones, get rid of them, and that's how you save labor in the seed orchard planting. Is you're not planting out everything. You get down to, let's say you get down from 150 to 25, you can plant out those 25 a lot farther apart.
Right.
And they might have a better chance, because then you have the competition effect. You have 150 trees, 1 foot spacing. Sometimes, just by way of what this tree grows slower than this one, it's going to get shade out and compete with it, and then it might have high resistance, but it's getting competed with. And so it's not a perfect assay.
My biggest fear of all the work that we've done is that the selection that we don't realize we're doing.
I fear it too, but we have to do it.
There's only so much we can do. Like it may turn out that the most resistant ones are the ones who grow the slowest, or whatever. And we've crowded those out just because of our methods. So any way that we can, having what we've learned, apply it to the Nankings, and then we can do with those to take them where we need it to go.
Yeah, I wouldn't stick with the 150. If you find that small stem assay is usable, or maybe we get to the point where it's cheap to genotype trees and you don't even have to do that.
Right, and we're not there yet, clearly.
Yeah, we're not there yet, but I would do everything in your power to not do that 150 tree block in a seed orchard, because that's a huge amount of effort. I'm seeing the effects of it later on. We're going through and culling all the trees. It's a lot of effort to rogue the trees.
Yes.
So
even in backcross, it's a huge effort because the trees are so large.
Yeah.
So
the cost of genetic screening has dropped tremendously since when we first knew how to do that sort of thing. You say it's around $60 to $100 a tree now. What do you expect in the future? Is that going to continue to drop considerably or not?
It depends on somewhat on what we discover in terms of the complexity of blight resistance. If we could actually narrow down a few very highly predictive regions of the genome for selecting for resistance, then we could target the genotyping. We don't have to do like 20,000 markers genome-wide. That's a lot cheaper.
Right.
So that would be the frontier, is like getting very confident in our predictive results for a small number of loci. The cost of sequencing is going down a lot, but that's going to be, the sequencers just generate piles and piles and piles of data, and it might be overkill for, it's useful for generating reference genomes, but it's maybe overkill for like a quick screen of resistance. So that's where the frontier is, narrowing and mapping the genes. Yes?
I have a comment and a question. The comment is that it's clear to me from all that you've said today and from what I've learned about planting trees and seed orchards that we do need, and whatever we do need to be planting a lot of seedlings in containers, whether we do small stem assays or not, because it does not work to plant trees at 1 foot spacing in succeeding years, because the competition really gets wrecked.
Yeah.
my question is the universal commingler hypovirus sort of on a coeval schedule with the transgenic
What'd you say? What was that?
release schedule? I mean, it's under review, right?
It's under review.
Is it on the same schedule? I mean, are we looking at 3 to 5 years, or?
Yeah, they are, Don Nuss and Mark Double, they're guys at West Virginia, they're testing, inoculating Cricomectria cankers with this hypovirus and doing some testing on it, so I think they're sort of in the research, like proof of concept and showing that it doesn't have harmful effects, so I think it's probably in that range too. The 3 to 5 year thing with Bill Powell, for the transgenic tree, they haven't even submitted to the U.S. government their application. They're still trying to gather evidence because they don't want to submit an application and not have all the right research done to show that it's safe, and then the U.S. government says no. Once the U.S. government says no, then it's very hard to go back and get them to say yes later. So they're trying to be very careful.
And
so
is this peer-reviewed research they're doing as they do it?
What is it?
Is it peer-reviewed as they do it?
Yes.
Prior to being presented to the U.S.
They're trying to publish papers, show like, oh, no effect on growth, no effect on pollinators, no effect on fungi, beneficial fungi, no effect on bacteria, or other kinds of things that are living on the tree. They just want to show that it affects blight resistance, and that's it.
Is the removal of the hybrids that you're not going to keep best done by wood chipping and scattering the wood chips, burning, or some other procedure?
Yeah, you cut the tree down, take a stump grinder, or you rip it out of the ground. It's a lot of work. Yeah.
And then what?
Burn it.
Burn it?
Yeah.
Yeah, there are regulations that we follow with, what is it, APHIS? Or,
Yeah.
something.
This may be naive, but I wonder what precautions you are taking for culturing the bad guys, the root thing and the fungus, and then inoculating. Are we spreading that into the environment?
It's ubiquitous.
I know, that was the argument I heard at first, but
We don't move it across state lines, but I think that's still kind of...
Are we breeding more virulent strains of it with our activity?
I think we're probably spreading some nasty, the most pathogenic strains in our orchards, for sure. Like...
Right. Why do you say that with a grin?
Because you want the trees. I'm sadistic about killing trees, because I want to get to the best. And so, yeah, we are inoculating with highly pathogenic strain and trying to put a very stringent filter on our backhouse hybrids. Whether it's going to affect the Native American chestnuts around, I think that they're hanging out as stump sprouts. Yeah.
EP 155 come from Connecticut anyway. I mean, it's close, right? I think it's.
Yeah, like it's native around.
Yeah,
there's a whole like
it's a few
spectrum of pathogenicity of the fungus, and we just happen to be using one on the tail. But in nature, a bunch of these strains are about as pathogenic as EP 155.
Because if you think that the trees themselves would, without our intervention, which you said at the beginning, can recover,
I doubt it.
like the birch forest outside in Europe, are destined to come back to be the dominant tree. Is it the beech? No, I'm sorry, the beech.
Yeah, I mean, there's been a breeding program for they take these large surviving American chestnuts that lived in the core range of the chestnut blight, and they're still living as big trees.
Yeah, we have a lot of them near the Quabbin. We could identify hundreds.
I've planted them out. I've looked at a lot of the progeny, the crosses between these large surviving, and I'm not impressed with the level of resistance. So,
Almost every tree that we have brought to our program seems to die within a year or two after we get the nuts. I mean, they have a lot of trouble surviving, even the large ones. That one at Quabbin I showed you the picture of, that got nicked by a logger or something, and that one's, I think, got blight. So even your greatest trees seem to succumb.
Has there been any work done on combining, I think the question kind of alluded to it, combining the Graves and Clapper and Mabry and King and getting improved resistance?
Well, you guys are doing that, aren't you?
Yep. We did a we did a program this summer, which I believe replicates one you're doing in Meadow.
Yeah, so the idea is, okay, Graves has a different resistance genes than Clapper. When you cross them together, you might get more variation. And there might be more, what we're hoping for would be you would see more trees on the high end of resistance spectrum. Even though you have to screen more trees to get to find those highest resistance trees, you find that there's a longer tail, I guess, in the distribution of resistance. That would be the hope with crossing these sources of resistance. But we still have, you know, John and Brian Clark made these crosses between BC3, the best Graves and Clapper, and they did Graves by Graves, Clapper by Clapper, and Graves by Clapper. So we should test that experimentally. So there's still science to do on that.
This is a question for those of us who like to go out into the forest and discover new trees. You said something about collecting leaf samples from a number of different trees. For example, what would the numbers be? Would you want like 50 different leaves from 50 different trees that you might find growing out in the forest? Would that be a good sample with the geographic coordinates?
Yes.
Or would it be like 50 leaves from a single tree with the geographic coordinates? Can you elaborate on that? What
Sure.
sort of input are you looking for data?
I think what I want is 50 to 100 trees dispersed throughout Massachusetts and Rhode Island with geo-referenced. If I don't have the coordinates, I can't go back and associate climate, environmental variables, soil with that, where you know, with the DNA sequencing that we're doing.
Should we seek out trees with diverse and extreme environments?
Yes. And I mean, if you find, you know, I think I'm going back and forth with this other tree geneticist at Virginia Tech. The idea was to go to, let's say, low elevation environments, high elevation environments, and replicate that. Sample like 20 trees from low, 20 trees from high in an area, and then go to another state or something and do the same thing, and just scan and be able to use like those multiple individuals to be able to do some statistical tests for differences. And then I talked to him last week, and he's like, I want dispersed. I want as few samples, but spread out wide. And I think that there's a combination of both that we need to do. So if you have, and I think I don't really understand or know what is the relevant, besides like being at the extremes of the range, like for cold tolerance, for example, I think is important for the northern climates. But I don't know like what are the dimensions of adaptive variation that we need to be testing for, but I would imagine low and high elevation or these different precipitation regimes or something would be important for adaptation in chestnut. So I think like if you had some ideas on what you think is important for adaptation based on your observations in the forest of where things are growing, then I'm open to sampling larger numbers of trees from single and contrasting environments, I guess. So you can help guide the science.
Jared, I would suggest, that's something, that's an easy thing to get our chapters to help with, but we should probably put together some sort of protocol for what specifically we're looking for, how to put the samples, how to package them, where to send them.
Yeah, we will, yeah.
And presumably that we need to identify them as American before we include them.
Of course.
When will we need this material?
I think like if we started, okay, so we're going to have a chapter science meeting in February. And we're going to talk about some of these scientific issues that we don't have consensus on, like how to do the sampling in American chestnut. Once we have done that, probably if we have raised some money by next year, I think by this next growing season, if you had a protocol in your hand and we could give you some guidance on how to go and collect, probably even as early as next year, you can start collecting those samples next growing season.
I see a bit of a cross between your request for us to put in save genomes and put them into orchards
and is looking for trees in extreme environments, that might be some university. I'm just wondering, would it be as simple as collecting nuts you found underneath the tree is a way to put those into local orchards to preserve the genome? And
Yeah, you might have to cross them, right? If they're isolated and they're not getting pollinated by other Americans in the area, you might have to actually do some pollination.
Well, unfortunately, the wild trees you find, as I'm sure you were well aware, generally have branches that are like 50 feet or 100 feet up.
I wonder if you could like take Tom Sielli, one of our regional science coordinators, took like a bag of, I don't know what he's doing. He took like a jar of pollen or something, and he threw it over a branch, and he was like pepper shaking pollen from Americans onto these other Americans. So I think there's room for like some ways of doing this like guerrilla style, you know, like without using.
We need drones with little clippers on them.
So one more thing, the whole thing about doing an assay, we get so many trees that say, I found American chestnut, but it's not flowering, and really it's not something we could do. But I get people constantly saying, I found it, here's a tree, blah, blah, get a sample, Kendra gets a sample. But it's understory. Is this the type of tree that could be valuable in an assay within the state, or does it have to be flowering and large?
For the diversity analysis, you don't need to be flowering and large, all you need is leaves. For the germplasm conservation, we obviously need seeds.
Right, it's a different thing.
Yeah.
Okay.
Yeah.
I have a question, but I don't know who's the best person. Anyone in the room can answer. I know in Massachusetts there are some forest management plans that are going into forests and thinning out the trees, taking out the worst, and trying to do forest propagation and all that. But in the process, they're planting chestnut trees. So I'm trying to figure out, is that a worthwhile endeavor, or it doesn't?
Is it like American chestnuts, or what are they planting?
Well, I'm assuming it's, and I don't know the specifics, maybe you know, that they're a backcross variety.
Was it?
Were nuts that were,
Was it us?
that were the nuts received from us, or you don't know?
I think,
Or were we doing it?
So very often, we produce a lot of nuts, some of which we can't use in the program. So there are people, let's say in Fitchburg, there was a Fish and Game Club that had a bunch of land cleared, and they said, do you have any nuts we could plant? And we had mud nuts, and they were genetically unknown, but some of them might have some resistance, so we give them to them, and they'll plant them, and they'll grow chestnuts, and a lot of them will die, but some of them will survive, and they'll be out there in the wild, and who knows what's going to happen. You know, it's like a huge seed orchard that you kind of roll the dice on, and you might get something. But for a time, they'll take off, and you know, very much like Tabasco, where the tornado came through. There's places where what comes up is all chestnut, because they'll just race everything to the sky, at least initially, till the blight attacks them.
I think like this restoration effort, once we have a blight-resistant population, is going to be not just TACF, but like state forestry agencies, and we're going to need to partner with even just private individuals to put these trees out in a large scale. It's not going to be just, it's like such a huge effort to repopulate forests with a species. So.
A few more questions, and then we're going to have to move on, because we're going to do an orchard tour and very soon. But please.
I have in addition to the response to the lady who's sitting there, that unfortunately our ancestors managed to bring into the United States a really diverse array of chestnut-like strains of wood. So they brought in Chinese trees, they brought in Japanese trees, they didn't test any of them for the blight that was on it. Those strains, you know, they brought them from various locations in those countries. The blight has been there a long time. It was probably very diverse there, too. Nobody knows these things. And then they brought it and trapped it all around. It survives in our forests to a smaller extent. I don't believe it reproduces as well as on chestnut. That's a really lovely breeding ground for it. But it is there in our forests all the time. Yeah, so we are not, our orchards compared to the forest size are tiny, you know. So,
yeah.
This is an aside, but what we're doing here for the chestnut tree we are about to start for Homo sapiens, and it's called All of Us. NIH just changed the name for precision medicine to All of Us. And you kids need to get your genetics done. There is one health provider organization around Boston that will start collecting for all of us. And it's going to be like a Facebook of the bioscience platform, so get your genetics done and plug into that and start doing your own research.
One more question.
So you can't use the current database because the mother trees are dead and you can't take samples from them. I mean, we have a huge, take all the mother trees that we've used, but they're all dead, right? So you can't use samples.
What I would like to develop with your help and the regional science coordinators is a map of every mother tree ever used in back crossbreeding and every mother tree ever used for germplasm conservation orchards.
Dead or alive?
Dead or alive, so that we know where we sampled. So we know where we sampled, so we can know where we haven't sampled, and then go from there as to like guiding further efforts and trying to seek out what we really need for. It wouldn't be useful to just continue sampling from the same clear cut over and over again for all the mother trees. You want to go and find these dispersed areas for additional conservation.
And the germplasm orchard is like New York does, where they take Americans that survive and move them to orchards.
Yeah.
Great. Thank you very much.